So I study ants, and I study ants because I'm interested in how complex systems work. So ants always live in colonies, and colonies always consist of one or more reproductive females who just lay the eggs. And then the ants, all the ants that you see walking around are sterile female workers. And the queen doesn't tell anybody what to do, doesn't give any orders, nobody directs the behavior of other ants. And so the question that fascinates me is then, how is it possible for an ant colony to accomplish everything that it does? And how is it possible that ants are such a successful group? There are 12,000 species, there are ants everywhere on Earth. Clearly they're doing something right. So I want to tell you today about work that I do in the desert in southeastern Arizona, very near to the Mexican border, near the New Mexico state line. This is my study site, and that's a rabbit, not an ant. And I've been working there now following the same population of colonies since 1985. And the ants that I study there are called red harvester ants. The species name is Pogonomyrmex barbatus. And this is a nest of a mature colony. So you see in the middle the nest entrance. And these ants are called harvester ants because they forage for seeds on a trail that they sometimes clear, sometimes not, through the vegetation that might go about 20 meters away from the nest. And they bring the seeds back and store them inside the nest. This mound is about 1 meter wide in a mature colony. So every year since 1985, I have mapped all the colonies on the site. This is a map from, this happens to be 93, so the site is not that big. It's about 250 meters by about 400 meters. There's a road. The boundaries are arbitrary. And every one of these little filled squares is an ant colony. Every colony has its name, which is a number. And every year I go back and find all the colonies that were there the year before, figure out which ones died, and put the new ones on the map. And by doing that, I've been able to track colonies through their life cycles as the colony gets older and larger. And because that development of an ant colony is important for the story I want to tell you, I want to now start and take you through the life cycle of an ant colony. So ants don't make more ants. An ant colony is not a population. It's actually a collection of sisters. But colonies make more colonies. And the way they do that in this species, as in many ant species, is in an annual mating flight, where all of the colonies in a population of colonies send out their winged reproductives. This is a male with wings. This is a female, virgin female with wings. And in these ants, as in most ants, you can tell the difference between males and females because the males have tiny heads and the females have much larger heads. And that's because the males live only for a couple of weeks. They don't live long enough to eat, and so they don't have much jaw musculature, so they don't need a head for what they do, which is just to mate. So there's a male on his way to the mating flight, and there is a queen, still has her wings, and she's mating. That's her abdomen, and she's mating with this male. That's his abdomen, his thorax, his head. And there's another male on top waiting his turn, and we've recently understood that because of the strange genetic system in this particular species, a queen has to mate at least twice, and they often mate many times. So after they mate, the males all die. And they're mostly dead by the next morning. They're kind of clustered underneath the bushes trying to get out of the sun. And the queens, the newly mated queens, fly off, apparently at random, drop their wings, dig a hole, go into that hole and start laying eggs. And in this species, the queen will lay eggs using sperm from that original mating for 20 or 25 years, and she'll never come out again. So here's a picture from my book, Ants at Work, showing the queen digging her hole on that first day after she mated. And here's a real hole. They go down about 18 inches the first day. They get down there, lay the first batch of eggs. Takes about 6 weeks for them to develop, and once they develop, the workers do everything for the queen. She feeds that first batch of larvae by regurgitating from her fat reserves, and then she never goes out again. So here's what a 1-year-old nest looks like This hole here is the nest entrance, and there's a pencil for scale, and here's the rock that names this colony. So these workers, these ants walking around, are ants that are produced by the queen who mated the previous summer. It's a 1-year-old colony. And here is a 3-year-old colony. So here's the nest entrance. There's the hole for the nest entrance. There's a pencil for scale. Even by the time the colony is 3 years old, they've started to cover the nest mound with little bits of rock. And here is a 5-year-old colony. And there is the nest entrance. And here's a pencil for scale. It gets to be about 1 meter across, and that's about as big as they get by the time the colony is 5 years old. So this is what colony size in thousands of workers looks like as a function of colony age in years. So it begins with 0 ants, just the founding queen, and she goes on laying eggs year after year, growing very quickly through years 3 and 4, until about year 5, when the colony gets to a stable size of 10 to 12,000 ants, and it stays at that size for the rest of the colony's life. Eventually, when the queen dies, there's nobody to produce more ants, and although the ants live for maybe another year, once the queen has died, eventually the colony will die. And it's at this point where the colony reaches its stable, mature size that it first begins to reproduce, that is, to send out winged ants to the population's annual mating flight. So I know how colony size changes as a function of colony age by digging up colonies of known age and counting all the ants. And it's hard to dig a hole in the desert, so we get a backhoe to dig a trench next to the nest. Here is a nest, and there I am standing in the trench. I'm wearing gloves because these ants have a really powerful sting. That's Stephen Kover, who's at the Museum of Comparative Zoology at Harvard and has a wonderful X-ray sense for finding the queen, which is really important because unless you find the queen, you don't know that you've found all the ants. That was a more recent version. We dug up a colony that had just moved. So here's what it looks like underneath. This is the ground. Here's a trowel for scale. And we find a whole mass of chambers surrounding the nest entrance. And that goes down, it's kind of cone-shaped, and it goes down about as deep as the mound is wide. And then there's another tunnel, which might go down another meter or so, which leads to a small chamber, and that's where we find the queen and all the brood have gone to escape the backhoe and us. So this is the flag shows, this is a nest that we kind of sliced down the middle, and the flag shows where the nest entrance was, and you can see that most of the chambers are around the nest entrance. And inside the nest we find the seeds that the ants store. They prefer grass seeds. Here's another picture of a tidier chamber, and often we find that the seeds are piled up very methodically. Here are the small ones underneath, and then these larger ones. Here's an ant for scale. And also inside we find the brood. So ants, like all insects, begin as eggs, and then they turn into larvae, look like maybe little caterpillars. And it's the larvae who do most of the eating. So most of the food that an ant colony takes in is actually consumed by the larvae. And after the ants are larvae, they become pupae, which corresponds to the cocoon of a butterfly. And when the pupa is mature, then workers come along and slice it open, and the ant comes out as an adult. Those are the ants that we see walking around. And once the ant emerges as an adult, it doesn't grow anymore. So the process that I think about is what I call task allocation. And that's the question of how, without any central control, any management, any direction, how the colony gets the right numbers of ants to perform each task as conditions are changing. So things happen to an ant colony. This is what the site looks like after the summer rains. And I think this is probably the function of the escape tunnel, not really to get away from me and my backhoe, but instead to have a place to go when the ground floods. And after the ground floods, there's a lot of damage to the nest. So the colony has to put extra effort into fixing the nest, into nest repair. And this is true for all of the different activities that a colony performs, that it has to adjust the numbers performing them. And I divide the activities that I see outside the nest into these 4 categories: foraging, collecting food; patrolling, that's supposed to be a magnifying glass because the patrollers search early in the morning; nest maintenance, building and taking care of the chambers inside the nest; and midden work, dealing with the refuse pile or midden. And the first thing you need to know about these different tasks is that the ants performing these different tasks are all the same size. So you may have seen some species of ants, actually the minority of ant species, in which the workers come in different sizes. But in this species, as in most ants, the workers are all the same size, so you can't tell what an ant is doing by just looking at it. So I want to show you this film that illustrates these 4 different tasks. And starting with the foragers. So the foragers leave the nest, and they go out in streams, which we call trails, although they're not very distinct trails, and I'll show you some pictures soon. They're more like, sometimes they look more like big blobs. They go out and they get to a certain point, they search around, and then they come back carrying whatever they found. As soon as they find a seed, or if they're really lucky, a termite, they bring it directly back to the nest. So what you'll see here is an ant, some ants carrying seeds going directly back. And the film is streaming really fast. I don't know quite why, but you might catch a glimpse of an ant carrying a seed.
This is not real time.
Well, the film is moving faster than, it is in real time, but the film is moving fast for some reason. Okay, the next activity is one that I'll tell you a lot more about, the one I call patrolling. So the patrollers go out early in the morning, and they walk around, not that fast, and they touch the ground with their antennae. So ants smell with their antennae. These ants, like most ants, don't see. So their universe is mostly olfactory. It's mostly about smell. And they sometimes stop and touch antennae with other patrollers that they meet. And they walk in this characteristic zigzag trajectory. And then after a while, they just go back into the nest. They don't collect anything. They don't do anything else. They go out, they smell around, they go back in. And the foragers will not go out until the patrollers come back. The next activity is nest maintenance. So the nest maintenance workers work mostly inside the nest. And they line the walls of the chambers with moist soil, and it dries to a beautiful adobe finish, so that the ceiling and the floor of the chamber is lined with adobe. And then they bring out, like these ants, they bring out the dry soil and put it down. They also bring out the garbage and put it down on the midden or refuse pile. So you see nest maintenance workers outside the nest only occasionally coming out, carrying something in their mandibles, putting it down a few inches outside the nest entrance, and going back in. So they're all mixed up in the stream of other ants, and all they do is carry stuff out. Then they go back in. Then the midden workers work on the refuse pile, which contains the husks of the seeds that ants eat, and also these little pebbles that they collect. And what you see a midden worker doing is, like this one, just picking up some little piece of garbage and turning it over and over in its mandibles. And we've recently understood that the ants are putting in a colony-specific odor into this stuff. They collect especially porous stones because it's a good receptacle for their chemicals, and they move it back and forth across the mound. They pile it one day here, another day there, then they move it back. So these ants, the foragers, the patrollers, the nest maintenance workers, and the midden workers, are only the 25% of the colony that works outside the nest. And once an ant starts working outside the nest, it comes in and out and into these chambers just inside the nest entrance, but it never goes back down deeper. So ants start out, we think that probably the youngest ants work inside the nest in brood care, feeding the larvae and taking care of the larvae. When ants that are working outside bring food in and put it down, then they just put it down just inside the nest entrance, and other ants come up from underneath, collect the food, and carry it back down again. And then they store and process the seeds. Then the nest maintenance workers are working to line the walls of the chambers and to carry the dry soil back out. And then the queen does nothing really but lay eggs. So even if she had the intelligence to somehow direct the behavior of the ants outside the nest, keeping in mind that the ants don't see, there is no way that the chemical information could get through this whole mass of chambers to the ants outside the nest in time to tell them what to do. Then there's this escape tunnel, but we don't think that the queen spends much time there except when somebody's digging up the nest. Then one of the really surprising things about this is that there seems to be a large group of ants who are basically doing nothing, who you could think of as reserves in the sense that if something important would happen, they would go back out and help. But in my now almost 25 years of watching these ants, I've never seen anything that brings out most of the ants. So whatever it is, if they're reserves, I've just never seen it happen. Or maybe they really are ants that just do nothing. And one of the interesting questions about the network of interactions that I'm going to be telling you about is whether this group of basically inert ants has some function that helps regulate the whole network. Did you have a question?
Yes, actually. So the students are supposed to find out if the same ant in one function, they're not starting across their functions. So is there a way of like tagging them?
Yes, I'll show you some pictures. The question is, how do I know which ant is doing what? And I'll show you that in a minute. So when I started out working on this question of task allocation, the question I started out with was, are these different activities interdependent? Does it matter to one group of ants what another group is doing? And so I did a series of experiments in which I changed the numbers performing one task and looked to see how that would affect the numbers performing another task. So for example, we put out piles of toothpicks right near the nest entrance early in the morning when the nest maintenance workers were active, and the nest maintenance workers carried the toothpicks away, and it took extra nest maintenance workers to do that, and I wanted to see if extra nest maintenance work would have an effect on the other tasks. And then we repeated these experiments using marked ants. We used to mark them with model airplane paint. It has to be hot, fuel-proof model airplane paint with these ants, and you could mark them according to task, and now we use a little bit more high-tech Japanese marker, but same idea. And we did this with ants that were marked according to task. So these are all blue nest maintenance workers, and here's a yellow patroller, and somewhere in here there's a green forager, which maybe you can see. I can't see right now. So the results of these experiments were that, yes, the numbers performing one task do affect the numbers performing another task. So the different groups are not working autonomously. For example, if I make a mess that the nest maintenance workers have to clean up, the foragers stay inside the nest. They become less likely to forage. And that was true for all the pairwise combinations of activities. And the next result was that ants actually switch tasks. So for example, if I put out extra food, then the patrollers will stop patrolling and go to collect the food. The nest maintenance workers will stop doing nest maintenance work, and they'll go to collect the food. The midden workers will stop doing midden work, and they'll go to collect the food. But not all transitions are possible, and this is how it works. That when more ants are needed to forage, like I just showed you, the patrollers will switch to forage, the patrollers will switch to forage, the midden workers will switch to forage, the nest maintenance workers will switch to forage. And when more patrollers are needed, and I created a need for more patrollers by creating disturbances that recruited out more patrollers. If more patrollers are needed, then the nest maintenance workers will switch to patrol. But if more nest maintenance workers are needed, so for example, I make a mess that requires more nest maintenance workers to clean up, nobody will switch back to do nest maintenance. And they have to get more nest maintenance workers from the younger ants inside the nest. So foraging is a kind of a sink, and all the other activities flow into foraging. And once an ant becomes a forager, it never goes back. And once I understood that, that meant that each ant must be making moment-to-moment decisions, not only about which task group it belongs to, not just am I a forager or am I a nest maintenance worker, but whether to be active right now. That is, if given that I'm a forager, should I go out and forage right now? When I first saw the results, I had thought it was possible that the reason that when I recruited extra nest maintenance workers, I got fewer foragers is that the extra nest maintenance workers were foragers that had switched over. But once I understood that once an ant becomes a forager, it never goes back to nest maintenance, that made me realize that what must be happening is that when extra ants are doing nest maintenance, that affects the decisions of foragers whether to be active. So it's not the same ants switching over, but each ant is making moment-to-moment decisions about what to do. And the most puzzling result of these experiments was that task allocation seems to be related to colony age. So it's related in this way, that older colonies are more consistent and more homeostatic in their response to perturbations, while younger colonies are more variable. Where by more consistent, I mean that if you take a group of older colonies and you perturb them or create a need for more work in some particular task group, do the same experiment over and over, you get the same result from different groups of older colonies. Whereas with younger colonies, a group of younger colonies will all respond one way this week and another way next week. That is, the variance within colonies, among colonies in a given week, is not any different from older colonies and younger colonies, but as a group, all the younger colonies will do one thing this week and they'll all do something else next week, which suggests that the younger colonies, the smaller ones, are much more sensitive to whatever is different between this week and next week. And the other difference is that the older colonies are more homeostatic. So as I ramped up the magnitude of the perturbations, the worse it gets, the more an older colony will just leave everything else aside and concentrate on foraging, so that the greater the magnitude of the perturbation, the more an older colony resembles an undisturbed colony. Whereas in a younger colony, the greater the magnitude of the perturbation, the greater the magnitude of the response. And what's surprising about this is that an ant lives only a year. I've lost my pointer, so you see the lifespan of an ant is only a year. So that means that the response of the older colonies, while it's clearly different, is not the response of wiser, more experienced ants, because the ants in the older colony are not any older than the ants in the younger colony. So something else about the organization is changing. Yes?
As new ants are born, do they go back to the baseline number of foragers, the number of other roles? And do new ants take on the role that a dying ant is leaving behind, or do they fill whatever need exists at that time?
Well, the flow is from the ants inside. So the question is, do new ants take on the role of the dying foragers? No, there's a sequence, and the new ants start at the beginning inside the nest and are recruited up into the pool of ants working outside gradually. So a new ant is in a different position in that stream than the dying forager.
So I guess what I'm asking is, if you put out lots of food and cause a huge influx of foragers, over time, as all those foragers die, does the nest get back to having a small number of foragers?
Yes. So the question is, over time, if new ants are recruited into foraging, will the numbers go back down? And the answer is yes, they will. Yes.
So you say there's a sequence. Can you manipulate the speed at which they move through the sequence?
Can you manipulate the speed at which they move through the sequence? Yes. If you create a need for more ants doing one task, you pull them out of the previous step into the next step. So, well, to go back here, so clearly something about the organization is changing as the colony gets older and larger, and since it's not the experience of individual ants, an obvious place to look is in the size of the system, because the obvious thing that's changing is that the older colony is larger than the younger colony. And so that's what got me started thinking about what kind of ant decision rules could explain these outcomes. And we know that the ants' decisions must be based in part on just what they encounter in their environment as they're walking around. Otherwise, there would be no ants at picnics if they couldn't notice the picnic. But it must also be based on their interactions with each other, or it wouldn't make any difference to the foragers what the nest maintenance workers are doing, and so on. So I'm looking for decision rules that are in part based on interaction. And I started to explore the idea that ants use simply the rate at which they interact to decide what to do. So the idea is, okay, I'm a forager. I expect to meet a forager every so often. And if I do, my probability of going out to forage stays the same. But if the rate at which I meet other foragers goes down, I become less likely to forage. And that in that way, each ant doesn't need to understand what's happening. Nobody needs to make any global assessments. Each ant just uses its own experience of its interactions with others to decide what to do, but in the aggregate, you would see the task allocation that we see, and you might expect such a system to operate very differently when the colony is large from when the colony is small, because if the ants have the same algorithm, the same thresholds in a small colony and a large colony, their experience, the rate at which they meet others, would change, because in a large colony, there's more other ants to meet. So if ants are going to use the rate at which they interact to decide what to do, they have to know who they're meeting. So you might wonder, well, how does an ant know the difference between meeting a forager or a nest maintenance worker? This is work I did with Diane Wagner, who was a postdoc in my lab. And we found that ants within a colony differ in smell. So it's well known in social insects that all social insects, like many other insects, are covered basically with a layer of grease. These are long-chain fatty acids or hydrocarbons. And that's how an ant or a bee or a wasp knows if another one is a nest mate, because they have a colony-specific odor. And what we found is that they also, within a colony, differ in odor according to task. And in particular, the ants that spend more time outside, the foragers, shown with the white bars and the hatched bars, the foragers and the patrollers, have more N-alkanes in their cuticular hydrocarbons, whereas the ants that are inside smell different. And then we did an experiment in which we took ants, nest maintenance workers, that had mostly been inside the nest, and we exposed them to the same conditions that foragers and patrollers experience outside the nest, namely high temperatures and low humidity, and we got those increases in N-alkanes. So it's what they do that makes them smell different. In the same way that a carpenter might get a callus on her hands, an ant, as she's walking around outside, comes to smell different. So the smell doesn't make them do something different. It's because they're doing something different that they have a different hydrocarbon profile. So I want to tell you about how the system works in particular with respect to foraging. So here's a picnic. As everybody knows, where there's ants, there will be a picnic. Where there's a picnic, there will be ants. It doesn't work the other way, as far as we know. That's a great example of the regulation of foraging activity. That is, how is it that the colony decides, first of all, whether to forage at all, second, where to go, like where the picnic is, and finally, how many foragers to send there. So I want to talk to you now about the details of the regulation of foraging in harvester ants. And to do that, I want to tell you about the particular challenge for harvester ants in the network that controls foraging. So first of all, harvester ants compete with their neighbors of the same species. And it works like this. This is a picture of the foraging trails of one colony. So here's the nest entrance. This is about 10 meters across here. And you can see that sometimes they go out in a kind of a line that looks like a trail. Sometimes they go out in a line that turns into a big blob. Sometimes they just spread out. And from day to day, a colony will go in different directions. And sometimes the trails of neighboring colonies meet. So now this is the entrance of one colony, this is its neighbor, and sometimes they happen to overlap. And so when they overlap, either one colony's foragers gets the seeds or the other colony's foragers gets the seeds. And sometimes they fight. So here's a picture from my book of an ant of one colony grabs the petiole, that's the segment between the thorax and the abdomen, with its mandibles, and they kind of tumble around for a while. And sometimes they just won't give up. And so the ant that has grabbed on eventually dies from desiccation, because these ants can't be outside in the heat in the desert very long. And the muscles of the jaws are so strong that they remain clenched even after the ant dies. The rest of the ant breaks off, and the ant that was attacked walks around for the rest of its life with the head of another ant clamped on. So the cost in overlap is mainly in competition for food, but also in the cost of fighting. And over time, just as trees in a forest compete for light, and it's hard for a young, small tree to get started near a large, old one, so in this population, by looking at the census data over time, we can see that new colonies are unlikely to survive near old, large ones. So the main premise of their foraging, regulation of foraging, has to do with competition for neighbors, with neighbors. So the next thing is that these ants have to spend water to get water. That is, they store seeds, and the foraging going out in the hot sun costs water because the ant dries out while it's foraging, and they get their water from metabolizing the fats out of seeds. So their foraging system seems to be based on the premise that it's not worth foraging, it's not worth going out to look when the search time is long, when there's not, food isn't available and you have to spend a lot of time outside looking for food. So it's not worth it to use up a lot of water to get in water. And the way that the food is distributed is patchy and also ephemeral. The distribution changes all the time. So they forage for seeds, and they collect seeds that are mostly distributed by wind and by flooding, not from nearby vegetation. So it's not the case that certain places out there are good, reliable sources of seeds and other places aren't. Anywhere is just as good as anywhere else, basically. There might be local patches, but they're unpredictable. And so any time that one colony searches an area, it has pretty much as good a chance of getting seeds as its neighbor. And the final thing is that search time is a good measure of availability of food. And that's because of the way that a foraging trip is set up. So an ant leaves the nest, a forager leaves the nest, it goes out for a while on the trail, and then it leaves the trail and spends a lot of time searching around. And eventually, as soon as it finds something, it picks it up and goes right back to the nest. So it goes maybe 20 meters from the nest with an average time of about 20 meters. And Blair Beverly, who was an undergraduate who did great work with me, but instead of going to grad school, he came to work at Google. He did a wonderful honors thesis in which he showed that trip duration is clearly correlated with search time. How long the ant spends outside is correlated very highly with how long it has to spend searching, and it's not correlated at all with how long it's spent traveling. So they walk so fast to the place that they then leave to search from that the time they have to spend searching is a really good measure of how long they're going to spend outside. So the idea is that the more food there is out there, the more available food is today, the less you have to search and the quicker you come back. And it seems as though that's pretty much the same everywhere on a given day, because what determines food availability is more whether it's rained recently and washed up the dirt off the seeds than anything special about a particular patch. So here's how they regulate foraging, given those constraints. The patrollers come out first. This is the hardest part for undergrads about working with me in the desert, is that if you want to deal with the patrollers, you have to get up at 4:30 in the morning because they come out right at, as the sun comes up. And they're the gatekeepers. They tell the foragers whether to go and which way to go. So the foragers don't go out, as I said, until the patrollers come back. So it's the return of the patrollers that signals the foragers that it's okay to go out. And I want to tell you about a field experiment that we did that showed how this works. Taking advantage of this fact, we knew that the foragers would not go out if the patrollers don't return, so we could collect up the patrollers as they're coming back, put them in a little box, and know that the foragers would not go out. So here's a picture of a couple of students collecting up the patrollers as they come back. And this is work I did with Mike Green, who's at the University of Colorado at Denver. And we did a field experiment that, in which we removed the patrollers. So we put the patrollers in a box so they wouldn't go back. And then we dropped into the nest little glass beads that were coated with the extract of the cuticular hydrocarbons of the patrollers. So little glass beads that smelled like patrollers. And here's a picture of the glass beads. And that was enough to bring the foragers out. So this shows the number of foragers in response to live patrollers, that's the control, and then two different extracts of petroleum hydrocarbons, the full lipid extract, and then the hydrocarbons, which are part of the lipid extract. And you can see that little glass beads that smell like patrollers coming in to the nest were enough to bring them out foraging, whereas little glass beads that just had the solvent pentane as a control, or glass beads that smelled like another kind of ant, the nest maintenance workers, didn't bring out the foragers. So this is how we know that ants are using just interactions with each other, and in particular, the smell of the other ants, to decide whether to go out or not. And it turns out that the onset of foraging depends not just on having patrollers come back, but having patrollers come back at the right rate. So we try dropping in the glass beads at different intervals, one bead every 180 seconds, every 45 seconds, and it's only when they come back at a rate of 1 bead per 10 seconds that the foragers will go out. So this shows the foraging response. There's also a foraging response if the beads are dropped in pretty quickly, but if they're dropped in too quickly, it's not as good as dropping in at the right rate. And of course, we got the 10 seconds by measuring the rate at which patrollers are usually going back at the time that they come out to forage. So it means that the foragers inside the nest are using the rate at which they encounter something that smells like a patroller, not anything the patroller does, not anything the patroller tells them, simply the rate at which patrollers come back to decide to go out. So it seems that for the foragers, the fact that the patrollers can go out and come back is enough to guarantee that it's safe to go out and try to forage. So actually, we were able to make the foragers come out sooner by putting in the patroller beads sooner at the right rate. So it suggests that what's happening to the ant is simply something like this, some kind of threshold, where the ant, when it experiences an encounter with a certain kind of ant, it has a response that has a decay. And if it gets enough encounters, often enough, its response is pushed over some threshold, and its probability, say, of going out to forage increases enough that they're likely to go out to forage. But if the encounter happens much, much later, then by that time, since this decay seems to be on the order of about 10 seconds, the ant has forgotten that anything ever happened to it, and the process has to start over to make it go out. So we don't know that that's how it works, but this is how we think it works. The patrollers are also telling the foragers which way to go. And we figured this out in some experiments in which we blocked off different trails. So this is a nest here's the nest entrance. And there are 4 foraging trails, and the entrance to the opening in the grass to the foraging trails, which extend out for another 20 meters or so, is marked with a pair of white flags. So it goes through here, and so on. And we took this foraging trail and we blocked off the entrance so that the patrollers couldn't get on there. And if we kept the patrollers from going on that little segment of the trail, which is maybe 20 or 30 centimeters, just on the nest mound, then the foragers didn't go there. The foragers didn't choose that trail that day. And so then we kept the patrollers from the mound, and we put down extract of different glands inside the ant, the Dufour's gland, the poison gland, and the solvent, and the Dufour's gland was enough to get them out there. So we found that the ants are telling the foragers which way to go, but they're only directing them towards a certain compass direction, but they're not directing them towards any particular food source. So the patrollers are saying, okay, it's safe to go out because we came back, and it's okay to go this way. And it seems as though the decision of the patrollers isn't based really on anything to do with food quality, but only that if the patrollers go out in the direction that the foragers went the day before, and if they meet the patrollers from the neighbor, then they don't choose that direction. So the patrollers are likely to avoid the directions in which they met the patrollers of the neighbor. And they're also, so if you can go out, you can come back fast enough, and you don't meet anybody from the neighbor, then that direction is okay, and that's the direction that the foragers take. So, yes?
How they found out the direction to go? I mean, when they come back, how they pass the information from the direction they go back?
By putting down this chemical on the mound. So they don't come back. If they meet the patrollers of the neighbor, we think this is what happens. If they meet the patrollers of the neighbor, they're less likely to take that way back. What we know is that if the more patrollers come back in a certain direction, the more likely the foragers are to use it, and that's because the patrollers, whenever they come back, they just put down some chemical. So if there's any process that makes the patrollers less likely to come back in a certain direction, it'll make the foragers less likely to use that direction. But they're not really giving the foragers any information about what's good or bad about that direction. So they're only saying, okay, we went out, this is the way we came back, it's okay to go out. So they tell the foragers whether to go at all and which way to go. Now, what determines how many foragers go? So it turns out that this is a second process that depends on the rate at which they interact. And here, it's not interactions between the patrollers and the foragers. It's interaction between the foragers that have just come back from a trip and other foragers that are coming in with food. And the way we figured this out was to take away foragers returning, first foragers returning with food, and then unsuccessful foragers. And if you take away foragers that are coming back with nothing, it has no effect. But if you take away foragers coming back with food, it slows down the rate at which more foragers go out. So the foragers go out on their first trip because of the patrollers, they come back, and then the probability that they go out again simply depends on the rate at which more foragers come back. Oops. So this makes sense from the perspective of the colony, because the rate at which foragers return is a measure of the availability of food. So the more food there is out there, the more quickly the foragers are going to come back because they won't have to search so long. So for a forager inside the nest, the rate at which foragers are coming back is a measure of whether it's a good day to forage. So they don't have to know anything about that rate, they only have to respond to successful foragers coming back. And we found that they respond really surprisingly quickly. So here is the cumulative forager outflow in one colony on a particular day. And this is the same thing per 10 seconds. So these are the same data. And at this interval, we started to collect for 2 minutes the ants returning with food. And you can see that within just a couple of minutes, the rate at which the foragers go out again starts to slow down. So they respond within minutes to a very small change in the rate at which foragers are going in. They're really surprisingly sensitive to fluctuations in the rate at which foragers are coming back. So returning foragers removed for 3 minutes immediately led to a decrease in the rate of forager return. So this was surprising to me because, after all, what they're collecting is seeds. The seeds are not going to go anywhere in 2 minutes. So these are not like the opportunistic ants that you see in your kitchen around here. Like the second that you drop a crumb on the kitchen counter, the ants are there. These ants are foraging for resources that just stay in the ground until somebody finds them. And so it's really surprising to me that they have such a fine-tuned system that responds so quickly to changes in the rate of forager return. And I think that it may simply be a function of the decay rate in their memory. That is, if foragers can only remember something for about 10 seconds, then they can only respond to something that happened very recently. So that this very quick adjustment of forager return rate is actually a byproduct of the memory, the decay in memory. So the fact, though, that it is so fine-tuned means that they can actually adjust forager outflow very well to food availability. That is, when it takes a long time for ants to find food, then that decreases the rate at which ants go out, and they can adjust quite carefully to food availability. Now, what determines where the foragers go? Remember that the patrollers are just directing them out in a certain direction. Another thing that, so a forager comes out of the nest and takes one of the trails that the colony is using that day and then goes off somewhere to search for seeds. And the other thing that Blair Beverly found out is that each forager is returning over and over to the same location on successive trips. So the forager leaves the nest and goes out somewhere, and what he did was to mark ants individually. So we had a 3-color system, a different color on head, thorax, and abdomen, actually 6-color system, but 3 places to mark. And so he could follow individual ants and found that the different destinations on different trips for the same ant was within about ½ meter. So the ant then, for the perspective of the forager, the ant goes out first thing in the morning when the patrollers come back at a certain rate. It goes out, it finds a certain place, and then it goes back to the nest once it's found something, and then it waits to go out until it encounters ants returning at a certain rate. Then it goes out again, it goes back to the same place over and over throughout the day. And what this means is that from the perspective of the colony, the return of foragers from one direction is stimulating more foragers in another. That is, there might be ant number 1 who's going back and forth to here all day, and ant number 2 is going back and forth to there all day, but the return of this ant from foraging here is going to stimulate an ant to go out over there. And so the system only works in a situation like this one where the food is distributed pretty heterogeneously but unpredictably around the environment, and a good day to forage in one direction is pretty much a good day to forage in another. So it's a very finely tuned recruitment system that doesn't recruit them to any place in particular, but simply adjusts the outflow of foragers for the whole colony according to the success of any particular forager anywhere. So it's the regulation of foraging by interaction rate, but it's not by recruitment to any specific location. So unlike some species of ants that are probably more familiar to us that make pheromone trails to particular locations, here they have an elaborate recruitment system in which there is no information about any particular location. So I want to end by just outlining the big questions that this raises for me as a biologist. One is the question about stochasticity, then the questions, which are the big questions for any biologist, about development and evolution. The question about stochasticity is when you consider, so this is the average response of 40 colonies to removals for 2 minutes, and you can see that overall there is this variability in the rate of foraging, and then after removals it declines. But look at the error bars and think about how for an ant it can't be that the rate at which ants are coming in is very, it's extremely variable. For the ant that is using this rate, the input that it's getting is extremely variable. And then ants are ants. You know, no ant is making any complicated calculations. They can't do a lot of very precise averaging or anything. And their response is not fully deterministic. So if you watch any ant, you will quickly learn that no ant ever does exactly the right thing every time. So what's really interesting to me is that with such stochastic input and such imprecise response on the part of the ants, that the system manages to work so well. The developmental question in biology is the question of how this whole process changes as the colony gets older and larger. And that developmental question is really a scaling question. How is it that network size affects function? What's the relationship between the size of the colony and these dynamics? And the evolutionary question is the one that arises when you realize that colonies actually vary in foraging behavior. Some of them are much more sensitive to changes in the rate at which foragers come in than others. And so one of the things that we're doing now is using genetics to identify which colonies have more offspring, to ask whether natural selection is acting on task allocation, and whether the colonies that are better foragers are actually collecting more food, having more offspring colonies, assuming that the variation among colonies is due to differences among colonies in the particular algorithm that an ant is using. That is, an ant that says, okay, I need to meet 10 foragers coming in per minute before I'm ready to go out to forage. In that colony, they're going to do a lot less foraging than in another colony where their threshold is lower and they say, I only need 2 ants to come in per minute before I'm ready to go out. So the differences among colonies in these thresholds would lead to overall differences that we see consistently year after year in how much a colony forages, and that might have implications for their reproductive success. And that would be how natural selection was shaping task allocation. So I want to end by trying to make a connection with what all this has to do with Google by showing an email I got from somebody yesterday who was visiting my lab from Taiwan, and he writes that I visited Google with my friends twice when I lived at Palo Alto. This interesting company have some particular ways to work. Their boss asked managers of every department not to limit engineers how to solve problems and what have to do. Just give them a direction or a plan. Engineers will make a temporary group and decide the ways to do. The process may be longer, but opposingly may be more efficient than the way that managers decide if they can. Each group seems to have no link between each other, but the whole company works pretty well. So I don't know if that's really true, but he goes on to say, in praise of Google, that it works something like the ant colony he observed in my lab, and it's like this. This situation is like the colony of harvester ants I observed in the small room. He means the lab where I keep my ants. I found that dead bodies and trash are gathered at some nook far away from the nest. Some individuals bring them out and throw away when they just walk for a short distance. But some individuals will pick up the trash when they suddenly meet it and bring it more far away. Some trash are brought back and forth between two nooks. Basically, the ants walk around carrying pieces of crud. They put it down. They carry it back and forth. It seems inefficient because the repeat steps and the longer process from nest to the trash nook. However, when I saw the nest, totally it also seems to work well. Each ant just knows something they have to do, but not an accurate process. I think that maybe all the tasks in a nest are finished in this way, and it makes the tasks more flexible, not a straight line. So I don't know if you guys walk around kind of carrying bits of garbage back and forth. And I don't know if Google really works like this, but it's clear that there are lots of interesting processes in nature that do work something like this. And I'd be really interested to talk with people about analogies between the ways that ants do things, and maybe not so much how you do your work, but how you perform the tasks that you're working on. So thanks very much. Yes?
You talk about how maybe ants only have 10 seconds worth of memory or so.
Yeah.
Presumably one reason is they're really small and they don't have much brain power.
Uh-huh.
If ants could afford to be bigger and have more mental resources, but everything else stayed the same, do you think that they would keep this model, or would there be differences?
I think that this model, so the question is, if ants were big enough to have bigger brains, would they keep this model or would they do it differently? Well, I don't know, but I think that what's amazing about ants is that they have evolved ways of doing things really well that don't require a big brain. So I think if you have a big brain, you think about what you're doing very differently. I mean, the key to the way that ants work is that no ant is thinking about what it's doing. No ant needs to understand what it's doing or why. And so what we see in the operation of an ant colony is the ways to develop a system in which each component doesn't have any global information. Yes?
So if the ants are blind and foraging during the day dries them out, why don't they patrol and forage during the night?
Question is, if the ants are blind, so they don't need the light, and it's hard work to forage during the day, why don't they forage at night? Well, other ants are foraging at night. They got there first. I don't know. Yes?
I wanted to ask, do you really think it's okay to study ants by destroying their homes and painting them with toxic chemicals and stuff?
Do I think it's okay to study ants by destroying their homes and painting them with toxic chemicals? Well, I don't like destroying their homes, so I don't do it very much. Is it okay? I don't know. But the toxic chemicals don't bother them very much because they do fine with the paint. So the ants that are working outside only have 6 weeks or so to live anyway. And as far as we can tell, having painted thousands of ants, we've never caused an ant to die sooner by being painted.
I've heard that painted queens will get thrown on, in Beacon, painted queens will get thrown out. Do you have an experience with that?
The question is whether painted queens will get thrown out. Well, I've never actually painted a queen. The queens that I have, say, in the lab are so precious that I wouldn't let anybody near them with anything. But the reason they're getting thrown out is because they smell differently. And so with all the ants that we paint, we have to let them dry out. And then the ants groom each other, and they put the hydrocarbons back over the paint. And so then they smell okay, and then they don't get thrown out. Yes?
So if it's a threshold function with the decay,
Mm-hmm.
determines when
forager ants come out at the rate that patrol ants are returning.
Yes?
How come they return, they don't come out at as great a rate if the patrollers are returning faster than expected?
So the question is why, when we added beads at 1 second intervals, why, how does that fit in with the threshold model? And that's a great question, and it doesn't make sense in terms of the threshold model. So the only situation in nature where the ants would all come back that fast is if something really bad happens. So you could imagine some extra rule on top of the threshold model that says, if they all come back too fast, this is a dangerous situation. But that's really me just kind of invoking an extra thing to explain the result that, as you say, doesn't really make sense in terms of that decay model. So I don't know. It's the short answer. Yes?
So if the patrollers sense any danger, what does it mean? They don't come back in the nest in order for the foragers not to go out? Or what do they do? How do they communicate the fact that there is a danger outside? Don't go outside.
The question is, how do the patrollers communicate that it's dangerous outside? Well, that's the thing. They don't communicate anything, but the rate at which they come back communicates something. If they all got eaten, or so one of their predators, their main predator is a horned lizard, if the patrollers all got eaten, or if it was such a windy day, as happens, that they all got blown away, then they wouldn't come back. So the rate at which they come back is the information. They don't bring any other information besides that.
So if they sense the danger, they have to choose between going back into the nest or not going back, because going back into the nest would mean, okay, go ahead for the foragers, right?
Yeah, the question is, what do they do if they sense a danger?
But they
But
could return.
I don't think that they're out there to sense danger. It's the fact of their going out and coming back that reflects this current situation. But they don't go out and say, wow, it looks like, a pretty good day, but I'm a little worried about that lizardy thing over there. They don't do that. So it's only the rate at which they come back which gives the information. Yes?
The ants that go and look around, and once they find something, they come straight back. How do they know to go straight back? Or how do they know how to get straight back?
The question is, how do they find their way back? It's the same question for the patrollers and the foragers. We don't know. In some related species, they use the plane of polarized light from the sun, so something about the direction of the sun. There are some ants that can see landmarks, but they're in the minority, and these ants, we don't think, can see anything. So we really don't know. One of the things that we're thinking about now is whether the ants might use the gradient of the hydrocarbons in the midden objects as a way to guide them into the nest entrance from the edge of the mound. But that doesn't explain how they get to the mound in the first place. It's clear that they partly use interactions with others, and you can see this in any ants that you look at, that, like the Argentine ants around here, that they, as they pass each other, they touch antennae. And if we mess with the foragers so that the foragers coming back don't have many foragers going out, they definitely take a much more winding trajectory back. And so it seems that part of what guides them back way far from the nest mound is the flow, is the interactions with others that they have on the way back. Because as they get closer to the nest, there's more ants. But we don't know all about how they do it. Yes?
I have two questions. One, when the ants are when they're coming back to tell the foragers that the controllers are returning, how do you know it has to do with encounter and it's not just the concentration of the chemicals, in the ambient environment being built up as they return? That would explain the decay function without requiring any sort of logic inside the
The question is, how do I know the difference between each ant's experience of encounters and the idea that that little pile of glass beads might create a sort of pool of pheromone of a certain concentration? I don't know that, except that I see them touching antennae, and I don't see the patrollers kind of clustering in a pile. But I don't know that for sure. I had a film that I was going to show, but I feel like I was taking too long. But you can see the. So maybe I'll put this on while the questions are going. I think this will work. Oh, I have to approach it from the other end. I mean, it may be that an ant. Oh, come on! Don't be silly. It may be that an ant somehow absorbs its chemical environment over a certain time, but it would also have to be quite time dependent or the rate wouldn't matter, right? Because if they, I mean, I guess that's another answer, is that if they were accumulating just the net amount of cuticular hydrocarbon smell, then the beads that we put in every 3 minutes should have had the same effect eventually as the beads we put in one every 10 seconds, and they didn't. So I guess that it could be some kind of accumulation thing, but it has to be very time dependent.
The other question was the inactive ants. Where do they come in the job sequence that I think was over?
I think that they're just a sink that some ants fall into there and never get out. That those ants were once younger ants that were down with the brood, and then they end up in that pool, and then they don't. They never do anything after that. But it's almost, in a mature colony, it could be almost a third of the colony. So one of the interesting questions is whether that might be a kind of a buffer zone, so that these dynamics that depend on the rate of interaction, it might help to have a group of inert ants that dampen oscillations or runaway effects in the rate of interaction and keep the system more stable. Yes?
You mentioned that when patroller ants go out and they find that patroller ants from another colony,
Mm-hmm.
they don't come back by the same route that they went there. As sort
Yes.
of a way of signaling that you shouldn't take this route.
Right.
How do they find an alternate route back to the net?
Well, what they look like they're doing is just meandering around. And so maybe they feel a little more meandering when they've met a patroller of another colony, and being a little more meandering tends, all of these are stochastic trends rather than, strict deterministic rules. So this is film taken from inside the nest entrance with a fiber optics microscope, and so you can see that's a nest maintenance worker coming up and wondering what the fiber optics microscope is. But eventually you see that they come past. And the idea is that the ants then, just inside the nest entrance here, are using the rate at which they meet ants coming back in from outside to decide whether to go out. And then soon you'll see some ants standing around outside, and you can see that they're very responsive to antennal contact where they smell the hydrocarbons of another ant. So the idea would be that a patroller is out there wandering around, and if nothing happens, it eventually wanders back. But if it meets a patroller of another colony, its trajectory changes a little bit, making it less likely to come back the same way and making it less likely to put down the chemical cue that when enough of it accumulates will get the foragers out there. So a group of maybe 50 patrollers can set the direction for thousands of foragers later on by some simple rule that links, that simply makes them a little more likely to avoid a place where they meet the patrollers of the other colony. And we see the outcome of this in that if ants of mature colonies meet their neighbors on a given day, they're likely to avoid it the next. So we know that they're doing it somehow, and that seems to be the way that they're doing it. Yes?
Do patrollers fight?
With other patrollers? No, we don't see patrollers fight with other patrollers, and fighting is a strange thing that happens on some days are fighting days. So there are days when they tend to, especially after a rain, and we think maybe the rain washes away hydrocarbon colony-specific signals on the ground, but there are some days after the rains when the ants go pouring out all over the place, and the foragers of neighboring colonies meet and fight. And there are other days when we see just as much interaction but much less fighting. So the probability of fighting per interaction changes very much from day to day, and it's very much through the whole population. And the only thing I can think of is that there's some climatic conditions or something that affect the spread or the volatility of some chemical that induces fighting. So some days are fighting days, some days are not, and I don't know why. Okay. Thanks. Thank you. you.